Everybody, it is October 13th, 2021, week 4 of Drexel's fall term. So it's our fourth GBM. Is it week 4?
Yeah, it is.
Okay, cool. Thank goodness. We'll do an overview of last week, and then I think I have progress, and then Alex has progress. He was here and he left, but he should be around. But we
I
can
need to discuss
yeah.
the paper today. How we're gonna do it?
Yeah. Because I would like to start that. I don't want to wait until the last minute to do that.
Yeah. Also, Ethan, we completed the EOSTS thing. Alicia and I, last night.
You completed what?
The whole paper, the Evo STS thing.
You did?
Yeah. So
I know you were bugging me about it. I
was
didn't think that the
okay. It's
results were all
okay,
that good.
bro. Something is better than nothing.
Okay, I know.
Yeah. So it's okay to be able to put on archive.
Yeah, I want to give it to Ed Kim first.
Yes, that was what we were thinking.
Yeah.
Where is Alicia? I almost missed my exam because of her.
Oh,
Yes.
well, paper's more worth it, I think.
I did a
Maybe.
50-minute exam in 30 minutes.
You're so cool. Awesome. So we'll get started here.
Good, Brandon.
Okay, yeah, Brandon muted himself. Good catch. All right. So let's go over the story. Story is we have this data set, a lot of good data, and I've been reviewing it this weekend and creating some scripts that are reproducible that we'll put on GitHub and that you guys can use if you download that data. Namely, we're looking at non-small cell lung cancer, and the imaging features are in CT and PET CT. We have genotype information, as well as RNA sequencing, and then clinical information as well. Data can be viewed right here. I've been creating just simple functions with some libraries I've found to calculate those features. So there is this really nice Python library called Pyradiomics, which is perfect for us. Can you post the GitHub in Slack?
I'll put that. I think it's there though.
Yeah. So there's this really easy library. It calculates about 160 features. And I just organized it into this calculate features function right here. So it just concatenates all of these different features. I was thinking of whether to go through these to try to understand them, but I mean, this has been validated, so I'm pretty sure we can just reference it. People who are interested in learning more about what these features actually are can dig into that. But I think that the figure that I've been throwing around and Safek and I have thrown around in the past explains it pretty well. It's just that figure from that one paper, this one, right? Kind of breaks down the static and dynamic features, and then these are kind of like first order right here, and then you get into the 3D, 2D features, and then higher order features, and that's kind of what this looks at. And there's other algorithms like grade level co-occurrence, grade level size zone, run length, that you can go into if you want to learn more about those. So from these features, we have about from this function, we have about 160 features from this. And I mean, I was thinking about this the other day because I understand that the best thing to do is to, I don't know if it's the best thing to do. Maybe you can comment on the subject of whether you just throw the image into a convolution neural network. I don't know if that's the best thing to do, or what's the downside of relying on these summary statistics as opposed to using the image directly. Because I hate to go backwards in time, right? I don't know if this is newer or if this is more reliable or this is less reliable.
And the problem, there are a few things with using CNN with medical imaging. So first of all, CNN could pick up non-medical aspects of the image and make a classification on the basis of that, because we don't know what it's classifying on. It could be true, but we don't know if it's going to stand true for all the possible inputs. And the second problem is people have used it a lot. So it's not novel anymore. So we need to come up with something new. Even if it doesn't work, we need to come up with something new. You could use maybe sparse coding. I don't know. But Dr. Kim said sparse coding couldn't work very nicely because it's a huge data set. And secondly, it's the images have multiple images tagged over each other. So the 3D imaging won't work with sparse. So that was a problem. So I was actually looking into what models can we use.
Do you have any ideas? I
No.
was going to just throw some simple models at it first. The reason I brought up I hope I'm not dancing around too much. The reason why I brought up the convolutional neural network thing is because a paper that was tagged in the Slack channel basically did that. It looks like they did some sort of ROI selection on a small part of the image itself. They didn't focus on the entire lung. They focused on a subset of the lung. And I want to show something else too. So the segmenting was something that can be tedious, but I don't want it to be. So I found this GitHub project that basically does automatic lung segmentation using UNet. I don't know much about UNet. I just know it works. So you can kind of see right here on the left-hand side, it labels the left part of the lung, or whatever the anatomical position is. This our left as green and the right as orange, and you can calculate features on either or, but for simplicity, I'm just calculating on all of them. And I think there was an example I posted in the chat of how well it worked. Yeah, right here. This was an example of applying UNet on our data set.
We could use a ResNet.
Well. Yeah, I'm going to use this for now just because it works.
We could use the Inception ResNet version 2 on Keras. It's pretty easy to implement. It's like a library, you just get it, because I've seen people using it on MRI scans.
Sure. Yeah, I just want to show this. This is just a DICOM viewer in JavaScript, and something we're going to have to talk about is, okay, we're at the point where we have features calculated, right, for a given DICOM image with the calc features function, and we're able to calculate that for a given region. So it calculates it only for the lung region of the body. It doesn't calculate for anything else. So it doesn't calculate the bed, it doesn't calculate the bones or the muscle. It only calculates for the lung region. The problem is that, you know, there's another dimension to this. There's so I'm scrolling in here, and we basically want a set of features per each patient, right? We want 161 features, or how many there are here, for each patient. So we have to somehow average across like relevant. I don't know how we want to do it. If we would average across the entire lung volume, or we would just pick the most representative lung slice. I kind of doubt that each lung is the same, like there's the same number of images per each lung. Like, I mean, this is one person, and so like if you start at the top of the lung here, like this is the top of the lung, and then you go to the bottom of the lung, I assume that, you know, not everyone has the same number of slices because everyone's a different size. So I don't know if we're gonna have to average or pick the largest part of the lung.
Ethan, so these slices are coming from top to bottom.
Yeah.
Right. So it's not about taking the largest one. It's about taking the one where you see cancer.
Okay.
Yeah.
Okay. So maybe it's
I
a
don't think
selection.
it's the selection, right? So one where the cancer is seen in the image, we take that slice.
Okay. So then we'll have to identify that one. I'm going to answer some questions. So the stage of the project, we've found the data set, and right now we're talking about the features, extracting features from the data set. So I'm using Pyradiomics library to extract features from the DICOM images. DICOM is a medical standard for medical imaging. It's how like MRIs and DICOM standard. If you go to dicomstandard.org, it kind of lays it out for you. It controls the image, the metadata, and it's what everyone uses in the medical field to talk about imaging. So the issue that we're dealing with right now is we're trying to figure out a couple of things actually, right? I looked into this weekend, getting the features for the RNA sequencing data, and it's not difficult at all. You just remove the non unique rows, and then you have to just apply some sort of like standard deviation shift or test statistic shift right here. But the main part, and I knew this was going to be the most challenging, is how do you get images out of the image? How do you get features out of the image itself? And what you can do, the method we're doing, is we're using this Lungmask Python project to segment the image. So we have a segment lungs function, and applies basically a mask. The segmentation acts as a mask, so you could kind of imagine that it's predicting. It basically finds the segmentation right here, and then you can just mask that over the original image just to get the region of interest, and you can give that to these features right here, these functions right here to calculate the features. But what I was talking to what I was talking about was I'm gonna get it wrong all the time. Was selecting the best CT image from each person, because we're going to want a set of features that best represent the cancer of each person. And we're using these features to predict the reoccurrence of the cancer.
I don't think they have cancer in this image.
No.
Not even in this.
The goal is build an AI that is able to detect and target cancer cells, not target cancer cells. The goal is to predict reoccurrence of cancer. So in addition to the metadata, in addition to the imaging data, in addition to the genomics data, we also have I think this is the clinical data right here. And the clinical data has a column for whether the cancer reoccurred and at what time point. So this is each person. These are their identifiers, and then there should be a reoccurrence column. I'm gonna probably miss it.
There's also a reoccurrence location as well, but I'll talk about that later.
Oh, I didn't know that. So there's a recurrence yes or no. I didn't see this actually. This is pretty interesting. And then there's the data.
And there's a location. And there's a location. That is.
So the idea is to build a model that'll detect A, reoccurrence pretty accurately, and B, maybe you could grade the reoccurrence, like reoccurrence that never happened, reoccurrence within 5 years, reoccurrence 10 years, reoccurrence 15 plus years, and see if there's some sort of conclusion that the features can explain that grading. But yeah, so over the weekend, my progress report is I created these functions, explored the Lungmask and radiomics, pyradiomics package, implemented a segment lung function, implemented the calc features function, and then also implemented the process RNAseq function. And I just want to emphasize that with this data set, we have access to about was it 22,000 our features here. Well, that's not. Oh, sorry. I just want to show what it looks like when it's clean. I think it should be about like 19,000 when I looked at it. It's a lot. It's over 15,000 for sure. And we might pick the most variable ones. So what I can do is I can, if we don't want to use all 22,000, I can take the standard deviation of each row and order it by the variance, order it by the standard deviation, and just pick the, 1,000 most, yeah, it's 19,000. I can pick the 1,000 most variable ones. That's a method that people often use to select features. The idea is that the variable, the most variable features, the most variable rows contain the most information. Also, we have
we might need to see the clinical relevance of the features in order to select.
Yeah.
So, if we get like a list which of them have the highest highest mean, basically, then out of those we could select the ones which are top priority and the ones we could drop out.
Yeah, these are all centered at 0 mean now. But yeah, something like that. Another thing you can do is you can, I wouldn't want to do this. You can actually look in literature to see what each of these genes correspond to, whether they're specific to lung or some immune response or something related to cancer. You can annotate whether they're tumor suppressors or proto-oncogenes. That would be helpful, because those are the two kind of categories that people look at first. Tumor suppressors are normally turned on, and then when they're mutated, they turn off, so they can't suppress cancer. Proto-oncogenes are generally turned off, and then when they're mutated, they turn on. One is dominant, one's recessive, just by nature. So next step for me on the programming side. So this is all on the GitHub page, right? We have a GitHub page, and I'm encouraging people to like take a look at it. It's not it. I'm encouraging people to take a look at it. I think I separated into features.py. This should be like almost updated. I'll push right now. For me, the next thing I'll work on is going to be, sorry. I want to organize this into a main.py with some, command line arguments. And then also we need to figure out a method for selecting the best image. And something like the best image per patient, the most representative.
If you want to use, there's this algorithm called refine nets, like units, and they are pretty cool. They give you the best image. They do the segmentation and they give you the best image.
I don't know. We don't need the best. We need the most representative.
Yeah, I mean, best as in the stack of their collection, the slices, the best slice.
Something else we have to keep in mind, and Alex, when you're looking at your clinical data, clinical features, we only want, there's only like a subset of people we actually want. There's about 130 of them, right? Because we only want the overlapping patients, and only 130 of the 211 patients have medical data, or sorry, RNA-seq data, and those are the ones we want. Does anybody have any questions about what I've done or what's available or what we still need to do. You'll hear more from Satvik and Alex, and they'll talk about different parts of the project. And I'm thankful that they're helping out with that, because this is it's a beast when you do it alone, but it's manageable when you can do it with people. I encourage more people to get involved too. But yeah, just to reiterate, right now, next order of business is selecting the best, the most representative image per person, and then it's just some software development stuff on my side to make this easy for people to reproduce.
Ethan, I have a question here.
Yep.
I need a clarification. So now, are we gonna use the image as well for the classification, or we just use the features combined with the genomics and clinicals?
I don't think we're going to use any of the I think we're going to use the summary statistics from the image.
Yeah, but no image itself, right?
No, we're not going to throw the image in. We're going to use the 161 features that we got right here.
Cool. So
because
I was thinking of using transformers.
Okay, I've never used transformers.
I've heard of the transformer models before.
It's if you look it up. Wait, I have this paper.
Look up transformers.
Wait, I think there's a. Oh, sorry. I think there's a Kara's implementation, if I'm not mistaken, somewhere.
Yes. I've posted this in the chat. This is for gene expression based classification, but I think it would work super good with us.
What are transformers?
It's like an algorithm. It's a new thing. They come up with new things every single day.
So like, it's a deep learning model, and it obviously it's deep learning, but like it takes each feature from the input data, and like they weigh it based on significance, basically.
Yeah.
Statistical significance.
I think. I'm not sure.
Yeah,
it's mainly like, used in it's used mainly for, in natural language processing and like
computer vision.
Yeah.
It's like weight, how important the input, like what part of input data is important. It's really cool.
I'll have to read this, and I encourage other people to check it out too. I looked into, and this is also a good paper to read, because it's the same thing. It's non-small cell lung cancer, and they're using probably a similar data set. That's it for me. It's up to whoever wants to go next. Alex or Satvik.
Okay, I'm just looking into a few more models and papers. 1 second.
No worries. Yeah, so like there's different goals of the project, right? The goal I'm focusing on is pre-processing the imaging data, which is kind of important, right? You need to have that data in order to apply ML. Alex is looking at the clinical features, and he'll be, performing an analysis on the other features as well. And I'm sure that, whatever he's working on can be extended to the features that I give him, because it's just going to be more numbers with labels, so that won't be too difficult. And then Satvik is, working on the paper and trying to figure out what models we're going to use. And, this transformer concept sounds pretty interesting. Never heard of them before.
Ethan, also, okay, wait, can I share my screen?
You may.
Okay. So for the correlation, this is also something I'm doing at my creativity lab. Can we make, instead of like a correlation matrix, can we make this, what do we call it, a graph, a semantic graph?
I can make a correlation matrix itself. I'm not sure about a graph though.
It's very easy.
What's your similarity score? Is the similarity score just going to be the correlation?
So this basically, this is semantic representations of the features. So think about this like this way. If I say the word cat and the word kitten, they would be really close to each other, like this of these group. And if I say the word dog, it would be far away. And if I say the word AI, it would be more far away. right? So this is the semantic difference between two nodes. And this is how you could see how these are clustered or they're spread out diversely. It's a nice way to form correlations rather than just a correlation matrix.
Sure.
It's easily you could do it. We can do it on R.
Yeah, so I'm going to answer some questions. Questions about the pre-processing of the data. Specifically, right now we have three kind of data sets. We have imaging, RNA, and then the clinical data, right? I'm working with the imaging. Previously, people used to take a, so I'm going to just, draw on a whiteboard. Let's pretend this is the area. The black part is going to be the image. Previously, what they used to do is they used to take a common region, so what's here in yellow, and they used to take all of this area and put it into a convolution neural network. Convolution neural network is a type of deep neural network, which is a type of artificial neural network, but it's really good at recognizing patterns. That's why it's conventionally been used in image analysis. What we're doing instead is we're taking each image and we're calculating these summary statistics. So we're looking at the size, the shape, the coarseness, other second order features about its 3D structure. And we're using those as individual features into our machine learning algorithm instead. It's to, improve. We're trying to be smarter about what features we're engineering and using. You don't just want to throw an image into a CNN because it's pretty expensive to do that. With the RNA sequencing data, so that data can be represented as a matrix where the rows are genes and the columns are the samples, so each patient, and each value is going to be a basically how much is expressed. And really the only pre-processing you need to do is for each row, for each gene, you need to normalize it. So you can take each row, subtract the mean of the row and divide by the standard deviation. That kind of, normalizes it and brings the mean to 0. And then you just remove the rows that are not variable. So if this row contained all zeros, I would get rid of this row right here. I'd get rid of it. For the clinical data, that data is pretty easy in my opinion. All you need to do is, the numerical data is just, it can be used outright, but the categorical data you have to encode. There are different encoding techniques. One type of encoding is called one-hot encoding. I mean, Alex will probably go into this one if you used it. The idea is that if you have categories like cat and dog, you can represent cat and dog with cat might be one vector and dog might be another vector. And all you need to do to encode that is to say, okay, the cat is signified by the placement of this one. And then for dog, that position has a 0, but the second position has a 1. And this becomes a one-hot encoded vector for dog. And then the edging in the graph that Satvik was talking about represents similarity between two nodes, and each node represents like an item. In the paper he was showing, they were proteins in like a protein network. The idea is that the closer the proteins are and the more clustered they are, that means they're more likely to be from the same network and be more related. Yeah, we're normalizing genomic data and performing encoding for categorical data. And then we're doing feature engineering for the imaging data, correct? Cool. All right, so Vec, you ready?
Yeah.
Cool. Yeah. I didn't add my name to the paper yet. That's funny.
Where's Zoom?
Yeah, I know. It always closes out whenever you close the Zoom. Whenever I stop sharing, it always, disappears.
Yeah, here it is. Let's go. Can you see my screen? Not this one.
Yes.
This one.
Yeah.
Okay, so first thing I would need is if you guys could put in your names, your college names, and put this here. This is Overleaf. This is a LaTeX editor we use to collaborate over research papers. This is a template that we have. I'll probably delete everything that is written in here. So I want to do it like this. Ethan, let me know if you agree with this, and also Alicia. We'll start with an introduction. We do a background study. I think we did that on our GitHub, but anyways. Then we go for a background exploration. Then we go into the methods. Into the methods, we talk about, first of all, the data set. Second, we talk about the segmentation, the pre-processing and the segmentation process. Third, we talk about the exploration part. Fourth, we talk about the application of deep learning. Then we come to results where we present all the graph, all the tables that we have generated and the actual results, how the classification came out to be. Then a section that I really want to add in this paper would be the ethics and biases related to this data set or this paper, which could also serve as the limitation to this paper, because we need to address them while talking about this whole AI and medicine thing. So we need to talk about that. And then we could end with a conclusion and future work, which could be done in this field, which is enormous. So yeah.
Yeah, that's a good outline. Something I like doing and something I'm gonna urge you to do, because you sound like you have it all in your head, is just put it on paper. So just outline each section. Yeah, yep. You have to do it now, but just if you do that, it'll make all of everyone else's work a lot easier, because then we'll see the we'll see the scheme, we'll see the story that we're trying to tell. But the way you outlined it sounds like a good story. I'm a fan.
But let me just write it up for myself.
The Latin we're using as a placeholder, that is just something people use in CS, or I don't know, in writing. It's just a placeholder. It's
because
gibberish.
We hate Microsoft Word. That's why we write in this.
Oh, she's asking about the Latin in the paper.
Oh.
It's just.
Yeah, I'll stop sharing. My MacBook is going to die.
Cool.
Yeah, I'm just writing it down.
The Latin does have, no, I'm just kidding. It doesn't have any meaning to it.
No, it definitely doesn't. Do you want to?
All right. Yeah, I'll share my screen. But yeah, if any of you, just like, especially new members, if you like, it's okay to be confused, because all of us are kind of at different skill levels, and that's totally okay. If you know nothing about AI, you're going to learn a lot from this. Like if you just like, you hear us talking about things like CNNs, like and transformers and all that stuff, like we'll explain everything, but also feel free to just look on it on your own. I wouldn't say you have to know how it works from scratch to use it. We're just using Python libraries for it. But yeah, this is the clinical data. This is the third portion of the data we're using. And it pretty much gives us roughly 38 different features of each of our 211 patients. But we're actually going to only use, I think, 100, right? You said it's
130.
130?
Yeah, patients.
Yeah, so I'm gonna have to select that, but that's part of data preprocessing. But yeah, just different things like their weight, gender, ethnicity, smoking status. They have some specific things related to the way they detect tumors, as well as the stage, as well as their diagnosis. There's just their pathological stage. I had to look a lot of this up, but yeah, different things. And what we're mainly trying to predict this, I'm not sure if we're doing the time series part, but we're definitely going to try to predict reoccurrence, as well as possibly location, although location is kind of sparse. There's like, I think like 80% of the data from this column is missing. I don't know.
It's not missing. It's just conditional on the first one.
Yeah.
Conditional on reoccurrence.
Oh, that's right. Yeah. Yeah. So we're pretty much predicting these columns as our target. So it's binary classification. we're doing, right? Or are we doing probabilities?
I mean, the
I
binary
mean, it's the
is
same thing. Yeah.
it's the same thing.
I assume we were also going to consider doing like recurrence over time, but we don't have the data for that. So it's just that one task, and we'll kind of funnel in the 3 different types of data into our deep learning model. You're going to say something, Ethan?
No, you're good.
And Satvik did talk about the ethics of it. And this is something that I brought up in the last meeting. So I'll actually put up here. The distribution of ethnicity and gender in our data is like not particularly representative. I don't really know, but I just wanted to hear everyone's thoughts about it. Like if you turn your head, you have this is not a scale of percentage. This is a scale of quantity. So we have roughly 98 Caucasian males, and we have various distributions, but we do not have any females who are Hispanic, Latino, African American, or Native Hawaiian, Pacific Islander. I don't know how that would affect our data. That there could be in infinite ways it could affect our data. So that's possibly something to include. Whether or
Alex,
not yeah.
I was gonna say, well, you can finish what you're gonna say. I was gonna say something.
No, it's just so whether or not I'm trying to decide whether or not we include this as a feature. We could either just remove it or just have a separate data set with the feature and without the feature. So you're going to say something, Ethan?
Yeah, from right here, I can't decide whether or not that's the case. And there's a couple good reasons I can't do that. The first good reason is, when you look at this, you think, oh, wow, we are over biased in terms of white men. But the thing that you didn't take into account is, okay, so what would be the best case?
Can I explain?
Yes, let's have to explain.
Okay, first of all, this is my book chapter. If anybody wants to read, it's on the ethics and biases in medical imaging. Just putting it up. So it depends upon where the collection of data set is happened. It has happened at Stanford.
And Virginia,
yeah, and both are pretty expensive places to go, right? And I don't think minority population of people from below socioeconomic backgrounds would go to Stanford to get their tests done, right? Or people from like different part of the country would go to just Stanford and get the results done. So it depends on the demographic of the city, demographic of the patient types. It depends upon the cost of the institution where the study is done. So this is a sample bias that we say, right? And it is due to the data collection, and it is not in the data. It is how the data is collected, and you have to deal with it. What could be wrong is the annotation bias, which could go wrong after retrieval of a data, in which the clinician does the segmentation wrong, or he takes wrong features. So the annotation bias is something in the hand of developers and the ML practitioners, and that we come around using units. And we also keep a check on units. This is not something we could call bias. It could be a downfall of the data set, and we could write about that, but not a bias that we could help with.
Yeah, thank you both for your input. See, even I'm learning a bunch of new stuff here, but yeah, this is just one
more thing I was trying to get at is you should represent it in terms of proportion, and then you should compare it to. I was asking what the best case is, and people would say the best case is everything is like equal proportion, but like that's
not realistic at all.
No,
yeah,
you should compare this to I'd say the national average for proportion. That would be something that I would think is reliable, or maybe California or something.
Cool. Got it. And also another thing I thought it was cool was the smoking year as well. It's kind of it's not normal. It's kind of like a hill. You could truncate it down to normal if you wanted to, but it's just pretty much the range of patients we have, so their ages. And you can compare it with this graph over here. They kind of look very similar. It's a little different format, but this is the range of our patients at their diagnosis, as well as just the year they stopped smoking. I just thought that was, I think these features are going to be correlated. Visually, I can see that, but that's kind of what I thought. And we also have this other feature. It says %GG. I have
%CG.
no immense, but
it's the percentage of their genome that is. It's the percentage of CGs in their genome.
Of CGs. Okay.
Oh, is that GG?
Yeah.
Oh, then I have no idea.
It's not like good. No, it's, yeah.
I thought it was CG content.
So yeah, it's just what I'm doing for those of you. This is part of data preprocessing, is like you pretty much want to analyze each of the features you have, and you want to understand them, because, for example, for this one, we have 65 missing values. So what do I do with those missing values? That's another thing that's going to come on based upon my knowledge of the data thing. But yeah, since this is a categorical variable, I just replaced it with the most common one here. There's probably smarter ways of doing it, but yeah, and then for continuous, you would replace it. I replaced it with the median, but then that also has its own issues.
Yeah.
So I got that. And yeah, if you scroll down here, the target variables, we got recurrence and then recurrence location, which we can have it as well output location. And then for the columns where it says no, I'll just add zeros in because it's not going to have a location. But yeah, that's mostly my visualization. I also wanted to share a paper that Suhani shared in the Slack. I think Ethan and Satvik already read through this, but this is kind of,
or this
what?
one.
Oh no, this is just this one. Yeah. So a group like is doing a kind of similar, has already done a similar thing to what we're doing. They're using a different data set, but they're we could use this as kind of just a jumping off point if we ever get lost. Let me try to share it with you all. Also join our Slack.
I think it's in the Slack. Yeah.
Yeah.
What was their genomic set like?
What was their genomic set? I haven't read through the genomic set part, but they used a
if you go down, there was a section. I want to see it. Gene data. It was RNA sequencing from the same platform. Oh, they looked at 5,500 genes.
Jesus.
From 88 patients. So ours is bigger than theirs.
And this got published where? Can you scroll up?
Where is this published?
We will have access to 130. That's our best case.
But what I just wanted to point out also was they also had clinical data right here. So they just use gender, smoking status, and cell type. For us, we don't have cell type. We have 4 different mutation types, which would help us. Ethan probably knows more than I do about this, but the mutation type also helps us determine that. And then also they did recurrence, but we could do a smarter version of recurrence, or just recurrence and location. So something like that. Yeah,
I wonder what happened if you try to predict all 3 simultaneously.
What do you mean? So like recurrence, location, and what else?
And recurrence time.
Oh, recurrence time. So date till recurrence.
Yeah, like if you graded them into if you bin them. I don't know if that would work well.
I don't know if, yeah, so date of recurrence. We do have that. We could try doing that. I mean, until Satvik picks out the exact models we're using, I was just gonna, once they pre-process the data, I was just gonna run through a couple just basic ML the ML estimators, but
yeah, That's fine.
Yeah.
Like linear, like logistic regression. You can do that first.
Yeah.
Oh, I have a list of which we could do. One second, I
Yes,
wrote down.
we're doing supervised because we have the labels.
Wait a second.
I don't think an unsupervised. I don't think this would really fit in the unsupervised way.
We could kind of cluster the images if we wanted to, but
you could use sparse coding on the genes to classify or to find a signature of reoccurrence versus non-reoccurrence.
So I'm also wanted to
genomics
mention for the clinical stuff, I'm gonna just work on Google Colabs. So the cool thing about this is that you guys can open this up on your own Google Drive account without having to download anything. So there's a way I can share it. Oh yeah, share it right here.
And then can you create like a functionized version of this? That's like create a function that's like called pre-processed clinical data.
Yep, I got that.
Okay.
So
you
just import in, it pre-processes everything. Got the function right here.
Yeah, because I'll want to include that into the features.py I'm building right now, just so it's perfectly reproducible. And I don't want I don't want like the Jupyter notebook on the GitHub because GitHub doesn't work well with binary files.
Really?
Ethan,
only text files.
I sent my the models I thought would be useful. I really want to see how it works with GBM. I'm really interested in because GBM Gaussian boosting model.
Oh,
yeah, it uses multiple trees instead of one tree. So because we have multiple features, that would be really cool.
Is that like an XGBoost kind of a thing?
Sounds like it.
Yeah.
Well, for everyone who have never seen these type of like models before, we'll go into them. I can explain most of them. I've never used linear discriminant analysis or GBM. I have used GBM
for my COVID thing.
Yeah. Oh, that's right.
Yeah. We could
explain them.
Yeah, we could host like this instead, like with these general body meetings, we could host like this sessions where if anybody has any doubts or want to learn something new, we could host up kind of a meet.
Yeah.
Maybe on weekends.
Especially like, if you guys like, if for those of you who just like join, you want to like getting involved, you don't know how to, join the Slack first of all, and second of all, just pick something to work on. If you want to like learn the very basics of like how to pre-process data and how to work with that, how to work with like tabular data, work with me. Otherwise, you can work with Ethan to pre-process the genome data. For those of you who have like biology backgrounds or are interested in that, you could work with Satvik on the paper and picking out the deep learning model as well.
The paper, and then the paper is going to be the difficult part. And then really what we're likely going to do is a grid search type of method to find the best model or find the set of models that best classify or can predict our target variable. Because every model is different, and there should be the best model for a given type of problem.
Also, if anybody wants the medical side of the cancer, there's this person Suhani. You could speak to her. She is working on the biological side of the cancer, how it spreads, and all of those things. So you could reach out to her. She sent a paper recently, which Alex was showing. So she could be a good resource.
Cool. I think that's all the progress. Something that I did on, I think it was Thursday or Friday, me, Alex, Savek, and Alicia, we met. No, Savek wasn't there.
No, I wasn't there.
No, but I met with Alex and Alicia and to just do some work and talk strategy. So I might do that again later this week. For me, it'll mostly be on the software side. So if anyone's interested, you can join on in, and we likely just talk in the general meetings just so people who are around can meet up. Yeah. Cool. That's it for me. Does anybody have any other questions about the project or, general question? Are these model types completely separate things that were developed in response to the same problem, or is there some kind of common philosophy to how people develop them? That's actually a really good question. I don't know. I think that they've been developed for specific types of problems, or not specific types of problems, specific types of circumstances. Like linear regression is old. I don't know like how support vector machine was introduced, but do you know, have any idea to that answer? Do you?
Yes. Yes and no. So what I know from my experience, I know this answer of how deep learning came into action from machine learning is that when the data is huge, first of all, deep learning takes more time to process data than machine learning, which is a downfall of deep learning. But when we train a machine learning model over huge data, there is not much increase in accuracy after a point of time. But regardless, on a deep learning model, it would keep on learning and learning, and the accuracy would go up. So that was one of the motive of translating from machine learning to deep learning when working on huge data sets. And as it comes to the models, it comes to the utility. And sometimes there's always a question when you start a research. Like there's always one primary question, what you want to do. Like, for example, in the GANS paper, it was addressed at, can we create something new which does not exist using a deep learning method? So I think, yeah, we have a sample problem question, maybe for a project they were working on. I don't know, but yeah.
Yeah, now after when you start to like look into, I don't care too much about the history of ML, so I know far less than other people do. I'm not a big history buff, but after working with ML and AI for a long time, you get a tool belt of algorithms that work well in different circumstances. And I don't know, it's like you're playing a sport. You just learn what works at a given point in time. Yeah, there's some resources. I mean, Alex pointed you to the data set, and this data set is a good example of something that that you should use AI on.
Yeah.
I'm also a little hesitant to use deep learning on this because we don't have a lot of samples. Just saying.
I have done. Oh,
you can talk to me. Just message me on Slack, and I can talk to you offline, or we can meet later in the week when we have a work meeting. Cool. Oh yeah, the segmentation was to actually find the region of the image that corresponds to the lung. We don't want the bones and the thing that the person's sitting on. We just want the area of the lung. That's where the features are encoded. Cool. Well, I learned something. Oh, here we go. So we remove a row of zeros in the genomic data set because that would be a feature with no variability and that's useless. Yeah, you do the same for clinical data. I'd expect that to not happen too frequently. The only really feature you'd see that with are the other case is when they're all different. Like if you have a unique identifier, that'll happen in the clinical data set. Like they're all given different labels or different like identifiers. So you'd also remove that because it's unique per patient. It doesn't really tell you anything about any underlying disease. But yeah, you would also do that in the clinical data if that was evident.
I think I cleaned out the paper. We are good to go now.
Cool. So
also, yeah, go ahead.
For processing 3 different types of data, you'd somehow encode them into numerical or categorical. I mean, recurrence or not is just Boolean, so that's 1 or 0. But then like a model that'll just innately turn into like a probability between 0 and 1. For the recurrence date or for the recurrence date, we can just, grade that into different categories or bin them. And then for area of recurrence, you can also treat that as a categorical. And maybe if there's 5 different occurrence areas, you just make it like a 5 different groups. And then I mentioned, having one model that predicts all 3 things simultaneously. That's good and that's okay in deep learning, but for other algorithms that isn't easy. So what you can do is just have 3 different models that predicts 3 different things, and that's fine. Each model is just learning something different. It'd be cool is if we something that would be cool. Oh, is binning. Yeah, binning is like if you have a set of time, you're saying, okay, one bin is for features that are, or for reoccurrences that happen, from 0 to 5 years in the future. Bin 2 is from things that happen from 5 to 10, and then bin 3 is from things that happen from 10 plus. If we had 3 different models, if we had 3 different models that classified these 3 different features, it'd be cool to see whether there's any like similarity between what they learn, like the feature importances, if there's any correspondence between the 3 models, because they're all learning some sort of mechanism of lung cancer.
Yeah, it could be. Yeah. I mean, like most of the models, they work on a tree-based kind of mechanism, right? So ADA boost, XC boost, GBM, random forest, all of them are like kind of a tree-based model. So I don't know, would be interesting to see.
Yeah, it would be cool.
But what I'm majorly focused on is this could be something like results we could compare to, but the transformers could be the main result that we're gonna show up.
Yeah, and that there's a Keras implementation for that.
Yeah.
Okay. Yeah. Good. Keras is a Python module or Python package for deploying like deep learning and artificial neural networks. And whenever anything is implemented in Keras, your life is so much easier. I've never built something from scratch, and I don't want to. I guess I'm a fake AI. I'm a fake AI enthusiast.
It's like dying in hell. Yeah. Hard coding a model. I've done that. Cool. I've made a unit, and it's like the company was working on the unit for like more than 2 years.
It's crazy. Cool. Well, that's it for me.
Also, quick note on the research paper, you are free to add in your name on the research paper, but also if you're adding your name, that means you have to contribute.
Yeah.
It's not just it's a good way to add names. No, we're not doing that.
Yeah.
So yeah.
Cool. I think if you have any questions, just stick around. I'll be here for a while to talk. But that's it. It's over. I hope they all leave. I hope you guys have a good night. That was so funny. You got it. Is there a possibility of false positive diagnosis recurrence? Yeah, it happens all the time. I mean, in the data, no, we're gonna have to take we have to take the data as the ground truth, but evaluating, false positive, true negatives in the data in the model is just a part of trying to improve the model. So that's something we're going to talk about in the future. Like what causes the false positives, what causes the false negatives. We'll talk about that, and we'll have to address that in the paper probably. You got it.
There's even everybody. So if I want to help out, I mean, my machine learning, like, yeah, no, it's definitely not spam. Like, from what you guys clearly display, but I'm like, my experience as far as like coding goes, I think it's pretty prevalent. But if I wanted to help out with just like processing the data and just getting it in a better format for like a model to use, how should I go about that?
Yeah, I would look at, I posted, yeah, thanks for speaking out and asking. I would look at the GitHub. So I'm working in the features.py on the GitHub, and that's kind of what I've been building up. Just getting familiar with that. And then we can talk about, incorporating what Alex has been doing, because I know Alex still has to create a function, right? You're gonna have to create a function of everything he's been doing in his Jupyter notebook and add that to the algorithm, to the features.py. But something that
is Alex here?
Yeah, he's right there.
Yeah, Alex is here.
Oh,
Something
sorry,
we shouldn't
I am on
overlook
another
too,
screen.
Brandon, that might be useful for you is, helping with the paper, because that's going to be a big task. It's always the biggest task.
Yeah, it's a lot right now.
Yeah, but I've been trying to make everything be as reproducible as possible. Do you have an external drive or anything?
Unfortunately, no.
Okay, the data set's about 100 gigabytes big.
Yeah, I've seen that. So
yeah.
Well, the pictures are, but yeah. The images and genomes are. But...
You can actually download. You can start downloading the data set, and you can just stop it after a while, I think, because you don't download the
samples.
Yeah, you can download individual samples off of the website.
Okay.
Yeah.
Yeah. Ethan, can you send me the analysis? From the segmentations.
Sorry.
Can you send me the segmentation analysis?
I didn't run it for all of the images yet.
Oh, you haven't?
No, I was waiting to do that until I developed more of the code.
Okay.
Because
I was thinking of making a graph. Can you send me the ones you have done? I want to.
I've done a singular. I did a singular image to test it. I'm not there yet. I don't want to because something I didn't talk about is how we have a manifest file for the images. So we're not going to segment every single image, because some images are just of the neck and of like the torso below the lung. So I have to figure out how. I don't want to generate more files than I have to. So I have to figure out how we're going to filter through that.
That makes sense. Also, Alex, I would say do not trust on Google Colab. keep downloading the page, the codes, because Google Colab crashes. It has happened with me.
It sometimes crashes, but I feel like the worst part is just losing the session. But I haven't had too many problems with it. I don't. What problems have you had with it? I'm curious about.
I have lost my whole notebook. That is what has happened. And I have, not kidding. Yes, and not kidding. I have got a $10 coupon from Google in compensation for that, because I bugged them for 3 months.
Wow. It's a high rate to get back.
Yeah. I mean, they said sorry in $10. I was like, okay.
Yeah.
I'll help.
Yeah, but Brandon, that's the best way to get started. And a lot of how I formatted the code in features.py is just like, because I'm trying to think about how we want to organize the data, and it's also from just having software development experience. But hopefully you can learn from it.
Yeah, awesome. So you're meeting on like Friday or sometime later this week?
Yeah, Friday probably.
Okay.
Friday or Thursday. It's tomorrow Thursday? Probably Friday. Tomorrow's
Friday.
Thursday. Yeah. Cool.
Yeah. You guys, good night.
Yep.
Bye.
I'll see you. Have a good night.
You have a very busy whiteboard, Alicia.
Yeah.
Yeah.
She's a lot of things going on.
Yeah.
A lot of things.
Our class is not a big deal for you then?
Well, I'm not in classes right now, but no, they're not usually in class. Like when I'm in when I'm not.
Yeah, we get it.
Yeah,
we get it.
That's the same for me. Classes are never a big deal. I want to do everything else.
Yeah.
Imagine taking classes.
Did you have a meeting with Wanting today, Alicia?
No, he canceled it. He had a meeting before, and it was running late.
Oh, okay.
I have a meeting with Dr. Osborne tomorrow, and I have to complete 4 chapters of that book.
Oh, tonight?
What is that?
Tonight. This is the neuroscience of intelligence.
Oh.
So, fun book. I have to read this.
Awesome. Well,
also, yeah. What's up with the USTS paper, Ethan?
Well, is it done?
It's done. It's almost done. One paragraph and it's done.
Okay, then tell me when. I'll post it on I'll post it on archive.
I uploaded my paper for the COVID, and it is still on hold for some reason.
That's it's fine. The conference proceedings was on hold for about 4 weeks before they accepted it. But usually individual papers they do pretty quickly. Like usually it's the next day. I was happy. I thought it was gonna be on hold forever, like the conference, but they said it was just an HTML page. I don't know if anyone else saw that, but
I tried looking at the papers. I couldn't even see them though.
it's because they're not all
you
uploaded.
cannot, Alicia. They're not uploaded yet.
The way this worked is that I uploaded the index to the papers, and now everyone has to upload their individual papers.
Oh, okay.
Yeah.
Because it's not peer reviewed.
Yeah. So like right here.
You can't like manually upload their papers, even though.
No.
So like this is like they'll upload their papers with this unique identifier, and then they'll take it to.
Why did you write my name, and rather than my? Why did you do it?
Oh goodness! I did that by accident. I guess to fix that. Oh, I'm actually kind of upset.
I'm like, okay. The paper is Satvik Tripathi.
I'll do that tonight. I'll do that tonight. Don't worry about it.
I'll let you know.
I just switched them. That makes me so bad. I rarely get anything perfect. Like for the number of things I do, I rarely get things perfect.
And I'll let you know when this is done. The LSTM paper, not the LSTM, EOSTS.
And then we gotta send an email because I want Ed Kim to review it because he,
Yeah. I would be in CCI tomorrow.
Cool.
Yeah. Oh, I want to show you. Is the recording done?
No, I can end it. Sorry, give me a second. I don't know where it is.
Yeah.